Thank you, Dr. Adler, for the introduction. It's truly my pleasure and actually a privilege to do this talk, especially knowing that more qualified people would have done probably a better job, but I'll try certainly my best. So I don't have any commercial relationships to disclose for this talk. I would have to say, however, that I did give a great deal of thought in putting this presentation together in my office. So this design actually was encouraged by our consultants and fosters productivity. Notice the absence of toilet paper. So this is a full-time accommodation. So in putting together this talk, what I tried to do was to look at relevant journals for the past year, calendar year, and I selected papers which I thought were important in furthering our understanding of the pathophysiology of inflammatory bowel disease. And when possible, I underscored the possible clinical importance of the new findings. Now, this is all in my humble opinion. Feel free to disagree. Tweet me, although Sylvester doesn't tweet, or put me on Facebook, that's fine. So I did not make the talk data intensive, and I'll tell you why in a minute. So this is a diagram that comes up in virtually every talk here at this meeting, and this is this Venn diagram of convergence of multiple factors, kind of a perfect storm scenario that results in the genesis of inflammatory bowel disease. I think what gets lost a little bit in translation is the importance of barrier function and how this relates to the pathogenesis of IBD. Now, I'm going to use each circle in the Venn diagram to kind of hold my own hand as I deliver the talk to stay focused on each topic. So I think a good hypothesis to begin with in this talk is that in IBD, there is an alteration of how the intestine deals with the microbial challenge that it faces every day, and this results in an auto-inflammatory condition. So in this setting, there is genetic predisposition of genes like NOD2 and processes like autophagy and reticuloendothelial stress that play significant roles, and that in the homeostatic condition or in the steady state condition, these processes are in balance. So we defend when we need to defend, and we tolerate when we need to tolerate. But in IBD, clearly this steady state is perverted, and as a result, this results in inflammation. So this is a schematic of the gut barrier that is composed of multiple factors, and of course there is the antimicrobial peptide secreted by Paneth cells in the small bowel, secretory IgA by B cells, the mucus layer, the epithelial layer, intraepithelial lymphocytes, and then immune cells in the lamina propria that detect and destroy invading bacteria. And to do all this effectively, we have sensing systems in the epithelium and in the lamina propria, including proteins like NOD2 and toll-like receptors. So in the last few years, there is another system that is gaining importance, and that system is called the inflammasome. So the inflammasome is an elegant structure, as shown here. It's very pretty, it looks like a spiral. And basically its function is to be a recognition sensor, and it's inside of the cells, and it responds to the presence of certain innate stimuli to generate IL-1 beta and IL-18. So in doing so, it needs to be stimulated by two hits. So the first hit will stimulate the maturation of procaspase-1, and this typically is a bacterial hit or a microbial stimulus, And then an innate factor such as ATP is the second stimulus. So in looking at the inflammasome in more detail, you will recognize that it's composed of different proteins. So one protein is an NLR, such as NLRP3. Then there is a component called ASC or ASC, and then there is the Cas-based molecule. And as shown here, you have a microbial stimulus, and then an innate stimulus such as ATP, glucose, and others. So these come together in order to stimulate the production of IL-1 beta and IL-18 in the appropriate setting. So depending on the context and depending on the concentration of these cytokines, you're going to stay in homeostasis, or you may develop chronic inflammation, or inflammation may resolve. So why is this important? So this is data that was presented yesterday by Scott Snapper at the meeting. And this is IL-1 beta and how it is important in the pathogenesis of inflammation in both mice and patients who are deficient of the IL-10 receptor. And please note in the horizontal axis the different stimuli that we use to stimulate immune cells in patients with IL-10 receptor deficiency. So you will notice that these patients just need one stimulus, LPS, in order to produce meaningful quantities of IL-1 beta. And this concentration of IL-1 beta increases further upon stimulation with a second stimulus, such as ATP. And this doesn't really happen in controls. So this is an autoinflammatory state in which IL-1 beta is important. And in this select group of patients, blocking the activity of IL-1 beta with molecules such as anakinra makes sense. And Dr. Rosen mentioned this in his previous talk. This can result in alleviation of inflammation while patients wait for a bone marrow transplant. So this strategy may pay off as we understand more and more about the inflammasome. Furthermore, the inflammasome produces IL-18. Now, IL-18 can serve different functions depending on the context. So in the steady state, IL-18 can favor the production of antimicrobial peptides, which control the ecosystem of the gut and foster the development and presence of diverse healthy microbiota. Now, these in turn can deconjugate bile acids like taurocholic acid and taurodeoxycholic acid to liberate taurine. And taurine can stimulate the inflammasome to continue producing IL-18. So this is a nice positive feedback loop to maintain health. However, during inflammation, there's a dysbiotic microbiota, and this dysbiotic microbiota, instead of producing taurine, will produce spermine. And spermine actually inhibits the production of IL-18. So it will not favor the homeostatic state. Now, what is interesting is that IL-18, in addition to its effects on the microbiota through the antimicrobial peptides, can also affect the density of Th17 cells, which are pathogenic in IBD, and increase the concentration of FOXP3 positive T regulatory cells, which will keep inflammation in check. So IL-18 can be good for you, but can also be bad for you. When IL-18 is in excess, it can in fact prevent the differentiation of goblet cells in the gut epithelium, and this can in fact produce damage to the intestinal epithelium by altering the mucus barrier. So it's one of those things of yin and yang, and we learned this the hard way with IL-17, where clinical trials in Crohn's patients using antibodies against IL-17 were not only not helpful, but were harmful. And we all thought that IL-17 was a slam dunk. So if we're going to target IL-18 as a therapeutic target, we have to be careful. Now, what this opens up is perhaps supplementing taurine in the diet can in fact have an anti-inflammatory effect. And wouldn't it be nice if this is one of the mechanisms by which exclusive enteral nutrition acts? Of course, this is now speculation. So switching gears a little bit, this is the obligatory slide where I show immune cells in the lamina propria. Now, to date, we all think about inflammatory bowel disease as driven by T cells, right? So here. Now, of course, T cells are important in the generation of IBD, but it's not the only lymphocyte that is involved. There is now increasing recognition that there is another population of lymphocytes that resides in the lamina propria, and this is called the innate lymphoid cells, or ILCs. Now, ILCs are of different types. For this part of the talk, we're going to focus on ILC3. So ILC3 is an innate lymphocyte cell that can respond to environmental cues, and these are present in the diet. You see the carrot and broccoli right there, part of a healthy diet, right? And these generate retinoic acid and aryl hydrocarbon receptor ligands, which can have immune modulating effects on these cells. And they also talk to other cells, immune cells, epithelial cells, and so forth. Now, the key concept here in understanding the function of these cells is that unlike T cells, innate lymphoid cells do not have a T cell receptor. So they have other receptors that can help recognizing all kinds of things, but they don't have a T cell receptor. They produce very important cytokines such as IL-22, which we're going to examine the potential importance in IBD. So this is one example by which innate lymphoid cells can have key roles in the genesis of IBD. So we all are familiar with thymic deletion of autoreactive T cells. And this occurs during development in order to avoid autoimmunity. So you see here in the thymus, there is the recognition of these cells, and then there's deletion. So it's interesting that innate lymphoid cells do this, but they do this for our own microbiome. So in the gut, what happens is that you have T cells that may react to endogenous microbial flora. And these cells can be recognized by innate lymphoid cells and deleted. In other words, they foster tolerance to our own microbes. Now, what's interesting is that, and I promise you paucity of data, so this is one of the few slides that has data. The bottom line here is that in looking at innate lymphocyte cells in the lamina propria of patients with Crohn's disease, and I direct your attention to panels D, E, and F, you will see that they are in lower amounts, and they also express less of the HLA-DR receptor, which is instrumental in deleting autoreactive cells to your own microbiome, to our own microbiome. And as the density of this HLA molecule decreases, then the amount of the number of Th17 pathogenic cells increases, and also the ability to react to bacteria increases as these cells are less abundant. So this is not important just theoretically, but also is important in IBD. So moreover, these innate lymphoid cells talk to the epithelium, and they talk to the epithelium through the generation of cytokines, such as epithelial-generated IL-1 beta through the inflammasome. And in turn, the innate lymphocyte cells will secrete IL-22, GM-CSF, and other key cytokines in order to crosstalk back to the epithelial layer. And this results in epithelial regeneration and survival, and the generation of mucus and antimicrobial peptides. So these are good cells. So is there a therapeutic opportunity here in having a novel target in IL-22? So this is now being explored, and there are a number of papers on this topic. So this is just 3 of them. So the IL-22 binding protein neutralizes IL-22, and T cells that are pathogenic in IBD make a lot of IL-22 binding protein, so that's not a good thing. There is a fusion protein that is now being developed as a candidate for treatment of ulcerative colitis that binds IL-22 to another molecule to prolong its half-life. And there's also data on the inflammasome and the generation of IL-18 and IL-22 as therapeutic targets for the disease. Let's switch now to the environment, and we all have seen this heat map looking at the increasing incidence of IBD over time, both Crohn's disease and ulcerative colitis in the world. And of course, in North America, we're very familiar with this. So I became aware of this study as I was searching the literature for this talk. So this is a study that is being done in about 500 volunteers in Holland, which are, these are healthy volunteers, extensively genotyped, and also there are a number of things that are being analyzed, including age, gender, BMI, and I love this, vitamin D, genomics, and their gut microbiota. So the idea here is to link environmental cues with the ability to react to immunologically. So let's look at study 1. So in the first study, again, the same 500 individuals, they collected peripheral blood, and they fractionated the peripheral blood into peripheral blood mononuclear cells, macrophages, and just whole blood. And they stimulated these cells with different molecules from pathogens, LPS and others, and then the readout was cytokine concentration, and they did a number of genotype studies as well as characterization of the microbiota. So one of the things that was discovered during this work is that there is seasonality with alpha-1 antitrypsin. So as we know, alpha-1 antitrypsin is a misnomer. What alpha-1 antitrypsin truly does is that it neutralizes neutrophil elastase and other immune cells. So they discovered that the alpha-1 antitrypsin actually has a seasonal expression, and you can see that here. So it goes up and down depending on the season. And going back to the theme of the inflammasome, the generation of alpha-1 antitrypsin was inversely correlated with the concentration of IL-1 beta, as you can see here. So this is provocative data. So we've been looking at vitamin D and saying, well, it makes sense. It changes with season. That's got to be the reason why, you know, the immune activation perhaps changes with season. But there is other unsuspected targets, such as alpha-1 antitrypsin, that may play a role. So we know that the alpha-1 antitrypsin is highest in February and lowest in the summer months, so inverse to vitamin D, that alpha-1 antitrypsin and IL-1 beta are inversely correlated, and that, for example, in other autoinflammatory disorders that involve the inflammasome, such as gout, there is an inverse correlation between the serum alpha-1 antitrypsin and the prevalence of gout. So, and interestingly, there was absence of direct vitamin D effects. So we don't know what the implications for IBD management are, but serum alpha-1 antitrypsin is now planted in my head as a possible thing to look at. So in going back to the interplay between the environment and our immunity, again, you know, the genes haven't changed, the environment has changed, prevalence of and incidence of IBD is higher and higher. So why? So we have Tregs, T regulatory cells. These are, of course, are FOXP3 positive cells that normally suppress inflammation. They are plastic, so they're not always good guys. You can make them into bad guys. And some conditions apply where the T regulatory cells actually start secreting inflammatory cells, inflammatory cytokines such as interferon gamma. Interestingly, in IBD, even though they're T regulatory, and you would expect them to be down, they typically are increased. So I came across this study that examined the role of salt in the diet in mice and the function of Tregs. So this is basically the bottom line in this study published in JCI. So peripheral blood mononuclear cells and T regulatory cells were transferred to immune-deficient mice, and they were put on either a no-salt diet, so no added salt diet, or a high-salt diet, which is HSD. And you will quickly recognize this is weight, so this is weight loss, that in the presence of a high-salt diet, these animals lose weight as a result of inflammation in the colon. Not only that, but the T cells in the presence of high salt will increase their activity and produce interferon gamma. So they become pro-inflammatory in the setting of a high-salt diet. So next time you eat sushi, don't add the soy sauce. All right, we're going to switch gears to microbiome now. Of course, this is a very popular topic. Who among us has not been asked about microbiome, right? My chair of pediatrics, who recently passed away, Dr. Larry Finberg, used to say that we're all covered by a thin layer of stool. Some is thicker than others, right? Okay. So in the dysbiosis that we've heard about, what is not really clear is what is the chicken and what is the egg, right? So is the IBD causing the dysbiosis, or is the dysbiosis causing the IBD? So I'll cut down to the punchline. It's probably a little bit of both. So this is a study recently published in Nature Genetics from the GEM study, Genetics, Environment, and Microbiome by Kane Cortoro and colleagues from Canada. And what you see here is, of course, the chromosomes, but the colored lines represent parts of the chromosomes that are associated with specific changes in the microbiome. In other words, it's not a casual interaction, but we influence the composition of our own microbiome through our genetic makeup. Moreover, in siblings of children with Crohn's disease, the changes that are present in Crohn's disease can also occur. So in the same household, you can have unaffected siblings of patients with Crohn's disease who have the dysbiotic changes in their gut. And one bug that we don't often hear about but is important is called Faecalibacterium prausnitzii. I practice that a lot. So there is reduced concentration of this, and we know that Faecalibacterium prausnitzii produces anti-inflammatory moieties that help maintaining homeostasis. In addition to that, in addition to the GEM study article, there is further evidence that genome-wide changes are associated with microbial composition. So even though we're colonized at birth by our mom's microbiome, our own genetic makeup is going to modulate the composition of that microbiome over time. We're now going to talk about big data. And before we talk about big data, I have a confession to make. I'm colorblind. So when looking at big data, this is kind of what I see. So this is Jackson Pollock, and this is called HOT9, okay? So basically, I like it. It looks beautiful. I don't know what it means, right? So in the next few slides, I'm going to present very briefly some big data, but I'll cut to the punchline very quickly. Big data, you saw it? This is the punchline. Oops, I have to go back to this. Okay, sorry. So what this means is basically that a low diversity in the microbiome is associated with the risk of developing IBD. This is the metabolome, so this is the products of bacteria, and the products of bacteria are also associated with risk of developing IBD. Sorry about that. And this is the same human study where the 500 samples are being collected, And what they did in this particular subset of the study is they looked at the effect of different bacterial products on the generation of cytokines. Again, big data, right? So as has been seen in other studies, this is the composition, a lot of variability. This is the metabolome, a lot of consistency. Now, in the generation of cytokines, there is diversity, though, and this is this graph over here. And I highlighted the TNF production of peripheral blood mononuclear cells relative to the composition of the microbiome. And as you can see from the sliding scale in this slide, the microbiome is important in conferring our ability to respond to certain stimuli in our response to cytokines. So it goes both ways. So genetics, microbiome, immunity. I know I'm running out of time a little bit, so let me skip over to this slide. So this is a different study in which stool from healthy controls, Crohn's disease and ulcerative colitis patients was transplanted into wild-type germ-free mice, and then the stools of these mice were transferred into a model, IL-10 knockout mice that has susceptibility to developing IBD. So here again, you see these are the donor samples, healthy control, Crohn's, ulcerative colitis. These are the recipient samples. And, you know, there is changes as they were transferred to the mice. And then the stool was transferred to IL-10 knockout mice. So what they showed was that depending on the subject, you got different results. And I think this is important. So for example, these are healthy controls. These are Crohn's patients. And this is histologic severity. And as you can appreciate, depending on the subject, you get different results. So this is important because it might have implications for a pro-inflammatory intestinal microbiome, our selection of patients and donors for fecal microbial transplant, and perhaps we should now be focusing on restoring not just the composition of the normal microbiota, but also restoring normal functions of that microbiota. I want to close with highlighting again the risk study, and Dr. Kugathasan spoke about the risk study yesterday. But in the risk study, there were about 900 patients with Crohn's disease newly diagnosed and were analyzed extensively to come up with a prediction model of susceptibility to complications, especially penetrating disease. So you saw this slide yesterday, but let me underscore the revolutionary nature of these findings. We will be able to reassure patients that their children, and this is at diagnosis, may not have a high risk of developing complicated disease down the line, and this should inform our treatment decisions and our talks with the families. So I brought you a semblance of the future. You like this? So I went with my son Dan to the Star Trek experience in New York, and here you can beam yourself in and you can beam yourself out. So just watch in a few minutes when I beam myself out, okay? So what are the take-home points? So we have an emerging role of the inflammasome in IBD susceptibility and pathogenesis. We have newly discovered targets that are sensitive to environmental cues such as alpha-1 antitrypsin in the role of salt in the diet. We may have IL-22 and its binding protein as new therapeutic targets. The intestinal microbiome and its metabolome and its interactions with host genetics and the individual variation are things to think about as we approach therapies. And also probiotics such as Faecalibacterium prasnitzii that are not in the Lactobacillus bifidobacteria category may in fact play important roles. And very importantly, as a result of the risk study, the ability to predict clinical outcomes in pediatric Crohn's disease is here and now. So to close, I would like to say that this Venn diagram has become more complicated. So not only do these things come together in a perfect storm, they actually talk to each other. So in thinking about the pathogenesis of IBD, I'm going to start thinking this way from now on, thanks to the advances in translational research in the past year. Thank you.